All right, welcome back to Chapter 3, Part 3. And what we're going to be diving into really kind of more of the fun stuff. We're going to look at biological molecules and how they interact. And you're going to get tired of me saying this, but structure and function. This is what we're talking about for biological molecules like various proteins that make up enzymes or other types of bioactive types of molecules. We're going to talk about those, but it's really, really interesting, and I hope you find it as well as interesting and fun as I find it. So, biological molecules recognize and interact each other. And this is very specific based on charge. We're going to talk about amino acids and the like, and they're made up of amino acids. And you think about it, we have really only 20 amino acids that make up all of life. It's sort of like keys on a keyboard or a piano, and you play the piano concertos and various things based on the notes. So you can construct various different types of music based on what you can do with those notes. Well, it's the same sort of thing. We have 20 amino acids and we compose all of life with a different arrangement of these in strings of amino acids to make protein. So we're going to talk more about that. But molecules with similar shapes can have similar biological effects. So I want to go over that first, but I want to introduce the concept of structure and function and specificity as we go along, and we'll get into it a little bit more. So amino acids have this basic structure. We have a carbon with a carboxyl group. It's sort of like a train, and I'm going to use that analogy later. These are the connectors from one boxcar to another. And the carbon and hydrogen region here is consistent with all amino acids. It's the R group, or the side chain that binds to this particular carbon, is really where each of the 20 amino acids become unique. We're going to talk about primary, secondary, tertiary structures of these proteins when they fold and all that, but we'll get into that. I just kind of give you the heads up. Redundancy and introducing things really help, I think, in terms of just being inundated all at once. I try to gradually bring it up. So, what is a protein? You can view this link right here at your convenience. It's a very good video on what is a protein, and you can watch that online. Uh, I think it's only 6 minutes long. So you just play the video if you want on your own, and if it starts to thread up here and play, we'll do that. If it's not, then I'm going to let you do it.
which link the amino group of one amino acid to the carboxyl group of another. A water molecule is released each time a peptide bond is formed. This linked series of carbon, nitrogen, and oxygen atoms is the protein backbone. Protein chains often fold into two types of secondary structures, alpha helices and beta sheets. An alpha helix is a right-handed coil stabilized by hydrogen bonds between the amine and carboxyl groups of nearby amino acids. Beta sheets are formed when hydrogen bonds stabilize two or more adjacent strands of amino acids. The tertiary structure of a protein is the three-dimensional shape of the protein chain. This shape is determined by the characteristics of the amino acids making up the chain. Many proteins form globular shapes with hydrophobic side chains sheltered on the inside, away from surrounding water molecules. Membrane-bound proteins have hydrophobic amino acids clustered together on their exteriors, so that hydrophobic side chains can interact with the lipids in the membrane. Charged amino acids allow proteins to interact with molecules that have complementary charges. The functions of many proteins rely upon their three-dimensional shapes. For example, hemoglobin forms a pocket to hold heme, a small molecule with an iron atom in the center that binds to oxygen. Quaternary structure. Two or more polypeptide chains can come together to form one functional molecule with several subunits. The four subunits of hemoglobin cooperate so the complex can more easily pick up oxygen in the lungs and release it in the body. Different visual representations of proteins can give us visual clues about protein structure and function. This space-filling diagram shows all of the atoms that make up this protein. This representation, called a ribbon or cartoon diagram, shows the organization of the protein backbone and highlights alpha helices. This surface representation shows the areas in the protein that are accessible to water molecules. Most proteins are smaller than the wavelength of light. For example, the hemoglobin molecule is about 6.5 nanometers in size. Hemoglobin is found in high concentration in red blood cells. A typical red blood cell contains about 280 million hemoglobin molecules. The three-dimensional shapes of proteins determine their function. The flexible arms of antibodies protect us from disease by recognizing and binding to pathogens and targeting them for destruction by the immune system. The hormone insulin is a small, stable protein that can easily maintain its shape while traveling through the blood to regulate blood glucose level. Alpha amylase is an enzyme that begins digestion of starches in our saliva. The calcium pump is aided by magnesium and powered by ATP to move calcium ions back to the sarcoplasmic reticulum after each muscle contraction. Ferritin is a spherical protein with channels that allow iron atoms to enter and exit depending upon an organism's needs. On the inside, ferritin forms a hollow space with iron atoms attached to the inner wall. Ferritin stores iron in a non-toxic form. Collagen forms a strong triple helix that is used throughout the body for structural support. Collagen molecules can form elongated fibrils, which aggregate to form collagen fibers. This type of collagen is abundant in skin and tendons. Learn more about the functions and 3D structures of proteins and other molecular machines at the RCSB protein databank.
I hope you enjoyed that. So the point behind this video is to kind of give you the feedback and I don't know, understanding of what I'm going to be talking about from here on out. And I thought I decided on the fly that it's probably best just to go through someone who talks a lot better than I do and provided really nice graphics. But it is in my slide set. If you ever want to go back and view it, it's excellent. It really will help us in this particular semester in biology, because we keep coming back to certain themes. If you've noticed, the chemistry we're constantly coming back to and building on that. If you have questions or don't understand something on the chemistry side, please reach out. I'd be glad to spend time with you to get that understanding. So the amino acids are strung together to form functional proteins. So as the video mentioned that, so we have a sequence of amino acids, and he mentioned, or the video mentioned 21. There is a unique alteration of a particular amino acid, but I usually don't worry about that one. The fundamental, there's 20, and these will arrange in a sequence in a chain. They're linked together using a simple hydrolysis, dehydrolysis type of reaction. So we can add or break those amino acids when we add them on through. We're going to talk about how that's actually done. But for right now, let's look at what the effects are once you strung them together like beads on a necklace. And then they form these functional proteins due to the different charges of that R group, as the video mentioned, and I mentioned, this carboxyl group and this nitrogenous group sort of is the front and back of each of the particular boxcars. So this would be the carboxyl, this would be the nitrogen group up front. So each one's sort of linked that way as it goes through. So I was trying to use that analogy. So there's the NH2 group and a carboxyl COOH group here, and then the R group can be any of these particular amino acids. And sort of the simplest one is glycine, which is hydrogen. But you can see some of these are rather bulky and large, and they have more designs, all of which follow the octet rule, by the way. But some of them are simpler, some of them are more complicated, and some of them contain like sulfur, like cysteine and methionine have the sulfur compounds. But if you notice how they're grouped, we have positively charged R group. The overall charge will be positive. There's an R group that has a negatively charged R group. There's a polar one that's uncharged R group and a non-polar and aliphatic R group. And you can just see that the sequences of those, of the particular carbons and how they're arranged, kind of dictate these groups. Here's a non-polar version: phenylalanine, tyrosine, and tryptophan. So we link these together. I'm not going to have you memorize these or anything. I just want you to look at overall the same basic grouping is here. There's the carboxyl group and the N group, and then the carbon that has the R group, which is in blue, which could be any of these particular amino acids linked together in the chain. So they come linked together for unique chemistry that make up a particular protein. So similar proteins would have very similar sequences of those amino acids, and it's that R group that dictates what charges they have and how they interact with others. So each is, we refer to the amino acids as the building block of a protein, and it's the composition of that side chain that really decides what it interacts with. And so you can see there's different chains, and we've got molecules of water that's kind of interacting, and that's something I wanted to make a reference to. It's assumed a lot of times these proteins really don't do anything unless they're floating in some sort of liquid environment. And I just thought that was interesting as a point that I can't overemphasize, because In liquid, then the charges, those individually weak but collectively very strong, can now actually have their effects in the water itself. So water is important. Let me give you a different diagram here. I show you what I'm talking about. So within a liquid environment, the side chains can interact with one another based on the position in that necklace, and they will fold as a result of the attraction of those charges. Because water is randomly moving, Brownian movement, we'll talk about that, but the water is just constantly bouncing into each other. It allows the protein to take on a natural shape, and that's due to those subtle charges that eventually fold together to form its tertiary structure. Now, the primary structure, as the video talked about, is the amino acid sequence. Then we go into the secondary structure, which either is a sort of a stepping of a folded pleated sheet, or starts to wound up as a circle type of structure. And that's fine, but eventually, as water gets in and allows the native charge of that protein to interact with itself, it will fold in a particular structure for you. And that's important, but the video talked about this structure, protection, regulation, contraction, and transportation, gave you examples. But this is a good way to think about that, having the different keys on the keyboard, or the different sequence of those amino acids, dictate the structure and function of what it does. So we of course try to deal with all the different things that these arrangements of amino acids can do. So we break them up into these sort of things that we observe: the structure and protection and regulation and contraction and transportation are different functions. And then we have examples of each of these, millions of them that do these sorts of things. And there is a handout that will be that is posted in the course resource area. What is a protein? It's a PDF document. It's also listed here, so you can download it. You can just click on the download button in my slide set. But this will provide you with a really nice printable 3D PDF document that shows each of those and gives you a nice little write-up and gives you some examples of each of those types of functions that we talked about. Now, there's one more thing I want to share with you, and this is something else you can play with. Now, I have to leave this the comfort of my little PowerPoint. You know, we get PowerPointed to death, and I apologize for that, but it is really the only way I can comfortably go over this material in some sort of logical fashion. So if you hit the link that was presented here, and I'll go back to it a little bit, but here's all sorts of various different types of proteins in this interactive. And if you want to look at enzymes, for an example, we can just pick out one of these enzymes. We're going to be talking about catalase. This is an enzyme. If you notice, all enzymes end with ASE at the end. ASE. So if I were to zoom in, here is something you can do. It's free. It's on. It works with pretty much any browser. You don't need Flash or anything like that. And in this part, you can rotate with your mouse, or if you have fortunate to have a touch screen, you can rotate it with your finger, and you can have different forms of way that's represented. This protein, you can see the solid form is here. This one is the cartoon. This is the spheres. This is the ball and stick. I particularly like the solids model version, or the best next thing is spheres. Now I can change from rainbow colors to what are the subunits. So you can look in catalase, and you can see that there's 1, 2, 3 different proteins. 4. There's 4 subunits in this catalase structure. In other words, this is a quaternary structure. These are individual proteins that are tertiary proteins that come together to form the quaternary structure, or this one that has two or more interacting proteins. Really interesting. So you can see the diversity of the sequences and what we can do with these building blocks. It just is mind-boggling. But you can go through any of these. You don't have to pick a particular one. Uh, Here's cytochrome, and again, I'll go with the chain version, and you can go in with your finger or mouse and zoom in and zoom out and look at these at your leisure. And it just shows you the diversity and shapes and function of a lot of these just based on how the amino acid sequences form in that necklace and how they fold to give us all of these machinery, the tools of life. And I would be remiss if I did not point this out, because this is really, really important, and it's fun to think about in terms of your experience in biology. And it's having these tools in 3D, because I'm going to show you things as we move on, the molecular nature of the cell, and I always present it that way. Yes, this is an introductory or principles of biology, but to give you the really good feedback of what's going on, I think you're going to enjoy the actual molecular part of this. So I hope you agree. The link is here. If you want to view it, all you have to do is click on the viewer logo here, and that will take you off to whatever your default browser is. And Chrome works, pretty much all of them work. I use Vivaldi. You can look that up. It starts with a V, Vivaldi. It's sort of like Chrome. It's based on the same foundation, but it doesn't have all the things that kind of slow it down. But anyhow, everybody has a choice. So to give you an example how specific proteins are and structure and function, you put a piece of chocolate in your mouth and your brain quickly registers, hey, it's sweet. We taste a little slippery and all that, but what exactly does this mean when a chocolate tastes sweet? And so what I wanted to do is to convey this with structure and function. And our tongue has certain receptors that, based on the shape and charge, it fits in a particular receptor that has that type of shape and charge, and causes a receptor to fire. Now I'm going to show you what that means, but it's just your neuro sensory units. Once it has something that fits into one of these slots, it changes its shape, it puts pressure on that nerve, and it fires and lets your brain know that, oh, you got something sweet rolling on your tongue. So let me get a little bit more technical than that. And you look at the human tongue has all sorts of taste receptors all over it, and you can see that it's not as pretty as that diagram that you had, but they have little folds and indentations, and the molecules like sugar, glucose, and that sort of thing would bind into those receptors, and you have a actual nerve once we get into the receptors. And this is getting down to the nitty gritty. So when the chemical, or like glucose or something, actually binds into the receptor and gets into those regions, we actually now have nerves that, once it binds into the particular region, wherever the taste bud is, the nerves will detect if there is a change in the shape. In other words, if it bound into that specific site that has charge and shape that's right, it puts pressure on that nerve and fires that nerve. And nerves look and work like this. Here we have down at the end, it's receptors, and we transfer from one nerve fiber to another. It eventually goes to the brain, you can see. And there is something interesting. I'll bring this up now, but we'll talk about it later. Nerves work by transferring an electrical potential. Then it goes through a region that's chemical, and it has to go through the synapse, and then it goes back to electrical again, which is really strange. And I'm not going to go much more into that, but I want you to think about that as we go on later. But here is where all the neurotransmitters and things, and we'll talk about that. But anyhow, once the receptor is modified, you generate a signal that goes through the electrical potential, and it fires that nerve and lets the brain know that it knows which one of those are wired to where, in terms of sweet, sour, and all those sorts of things, and then gives you that view, that mesh of information your brain knows how to sort through and say, oh, this is nice, and oh, this is sour, or that sort of thing. And as a result, the neurons fire, and the information through the synapse goes eventually to the brain. And then we can determine, oh yes, well, these receptors, oh, that was salty, bitter, could be a combination of these. See how amazing our brain sorts through this information and adds and subtracts and gives you a bouquet, I guess you could say, of flavors and experiences. The fat and the sugars and the salts and the sours and all that. And then you get this array of feedback, neurofeedback to the brain, and it sorts it through. Some of them could be very desirable. Some would say, oh, no, we don't like this, and that sort of thing. So, structure and function, and it's the interaction of these things to the receptors. And that's a kind of a complicated view, and I'm not worried about it. I just want to show you that we can look at this and not be scared by it. But what it's really showing is when something binds into a receptor, it changes its shape. Very much when you put in a key into a lock and you twist it, that lock becomes unlocked and it pops out and it changes it. So I guess that's the best I can show. Here's a free form of a protein, some receptor, let's say, the sweet receptor, and it binds to the receptor itself. And when it does, it changes the shape, and when it does is it rotates or does whatever it's going to do, and it causes the neuron to fire, and it lets the brain know that, hey, something just filled in that receptor that tastes sweet or whatever. So as we know, there's artificial sweeteners. We're going to talk about that, like saccharin and aspartame and those sorts of things. And if you notice, it says, wow, aspartame is 150 times sweeter, or saccharin is 450 times sweeter. Splenda is 600 times sweeter. How is that even possible? And if you've ever experienced sometimes somebody ringing the doorbell, and a very polite person will push it once and then wait for a while for you to come to the door. But if that person happens to be, problem, or I won't say we're a mixed company here, but a pain, and they start pushing that button, bing, 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 and you're just, well, the doorbell is actually becoming more of an alarm, but it's a lot more effective, I guess, when you keep ringing it to get your attention to come to the door than if you just push it one time. That's what these are doing. These molecules emulate the structure of the sugar charge and shape, but it persists longer in the receptor site, and so it's equivalent to it's like somebody keep pushing that little stupid doorbell, and ding, 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 we're ringing the bell, or whatever you want to say, and that's how we can get the sweeter like effect, because that normal sugar wouldn't bind in there but one time, ring the doorbell one time. But aspartame, which is not good for you, saccharin, which causes cancer, and Splenda, God knows what it'll do, but it just rings that doorbell so many times, your perception, your brain says, oh, this is really sweet, so you don't put as much on there. Unfortunately, these are chemicals, they're not actual sugars, and chemistry of this, your body will break it down. Even though it achieved the function of making you feel that it tasted sweet, the chemicals, however, have an impact that have nothing to do with sugar. So anyhow, food chemists do this. This was aspartame was actually generated by a chemical company, and a gentleman spilled a little bit on his hand. This is what I've been told. And he tasted it. Now, what idiot would do that? I mean, if you get a chemical in your hand, you don't know anything about it, the last thing you want to do is go and lick it. Well, this gentleman did that, and it tasted sweet, and he reported that to the management. And, you'd be amazed that a lot of findings, new discoveries in products and things that we enjoy, like tires. Believe it or not, there's this tire construction called carbon black. And I don't know, I shouldn't say this, but someone in one of the shifts urinated in the rubber making process, into the actual rubber making mixture machine, and as a result the urea actually changed or conformed the carbon that was in the tire rubber contents and made it stronger. Now, obviously this person had to fess up that he did it, but now that became a much better rubber. And we see these tires. Now, if you didn't listen to this video, would you have ever have known that? I mean, is that something you want to know? I don't know. I doubt it. But anyhow, I add that because a lot of times these types of things occur just by happen chance, and then they'll commercialize it now. So for us, sweetness is obviously a big important thing, and it just confirms the power of here's sugar. If you notice the structure, we know it's glucose, and then if you look at saccharin, it has one end of the molecule that looks really close to glucose, but because of all this other stuff that's on it, it's just a chemical that does a lot of things. This is actually broken down in formaldehyde in your liver, so enjoy your aspartame. It persists because it's a much larger molecule, and sits there and rings that doorbell, and you get that sweetness effect. And so it's the interaction and how long it rings that bell, or you constantly leave the key into the lock, however you want to look at the analogy. But I hope that conveys what it is. You can read about or watch a video on aspartame if you want to look at some of the history of that. I try to relate a lot of the things we talked about here to real things that hopefully that you can impress others, but more importantly that you use it to understand. Now, there's growth hormone. Here's a solids model, and the growth hormone is here, and then the receptor, you notice how it fits just beautifully into the receptor. When that does that binding, it changes the shape of that particular growth hormone receptor and signals the body to produce some hormone or something. It's a signal, and the signal occurs just like when sugar binds. It changes its shape, presses on a nerve, or causes a bind somewhere, and the nerve fires as a result. The same sort of thing in this case. The growth hormone is triggering now a cascade of effects that cause bone to grow, or the chondrocytes and things to signal that the fact that we're going to be adding more bone. Now, here's an interesting one. I like to use this. Now, I probably have a lot of junkies out there right now. Now, I'm not trying to make anyone feel bad, but every time you go to the gym, which I understand we probably can do now, you can go to the gym and work out. And when you work out, your body produces a natural chemical called endorphins. And it is a type of site when it binds to the endorphin receptors that makes you feel good. And it's sort of like what drugs do for various things. So it's a drug. It's a natural drug, endorphins. And what you do, if you can continue to do your workout for about 2 weeks or so, your body gets dependent on this motivation from endorphin. And the endorphins will bind, and you feel good. And then let's say one morning you're starting to be lazy and you don't want to go to the gym. Then all of a sudden your drug dependency on endorphins starts to kick in, and you say, gee, the last time I went to the gym I started feeling really good. And so you get out of the bed, a nice warm bed, and get clothes on and get in the cold air and go in the car and go to the gym or whatever you do, and you start to release the endorphins in your workout, and you go, oh great, and your body's all happy, and it gets those receptors where the doorbell's rung at the endorphin receptors, so your brain says, oh, life is just wonderful, and there we go. But there is something that mimics the natural endorphin, binds to the endorphin receptor, and it's morphine. And what that does, of course, when you give someone morphine that's in a lot of pain and is suffering and life is bad, then all of a sudden you get morphine and it binds that endorphin receptor, rings that doorbell, and your brain says, "Oh, life is good," even though we've got some pain going on. The endorphin receptors gives you a sense of well-being. It's release of various things that happen to do that, but all I'm saying here is chemically it looks very similar to the natural endorphin, and that's mimicry, but we take advantage of it with the drug morphine. Now, there's lots of drugs that we take for various things that do the same sort of thing. It has an effect on a receptor, and it causes the doorbells ring. So you just go in and you can see how similar the structures are. And then if you look at the receptor, the endorphin, the natural endorphin will bind and the morphine. Now, again, it's the business end. This part of the molecule has the same shape and charge as morphine, and it now renders the same sort of effect. Now, it may linger and ring that doorbell more often than, let's say, the natural endorphin, which I believe is the case. Morphine will break down. But anyhow, the summary, though, is the properties of a molecule, such taste, smells, or binding with other molecules, depends on the shape and the charge. Those things are really, really important, and it's within a liquid environment. Don't forget that. And pH and temperature and all those things can affect this, and so it's really important. Now, your book has another example. It's probably not as exciting as some of the other examples I've talked about, but molecular interactions in insect mating behavior is heavily influenced by the molecular interactions, and gypsy moths, for example, will fly as much as 2 miles. Boy, that's a heck of a motivation, because a molecule in the encounter fits into one of the receptors and tells them that the sexual receptive female is nearby. Well, the truth of the matter is perfumes and things like that, ladies, do the same thing to attract males and vice versa. So anyhow, but we'll leave it to gypsy moths because it sounds good. And so the receptor site is something called a pheromone receptor. And the pheromone, now I can have, I forget what it was. It was a big pen. It has an ink in it that I can draw on a piece of paper, just draw a line, and ants will follow that pheromone-like substance that happens to be in that blue ink. Now that sounds crazy. It's the same thing. It's like the pheromone. And the pheromone will bind the receptor, and the male gypsy moth can detect and track the female as far as 2 miles away as it gets that molecule. But the ants will follow that pheromone with the ink. I forget which is, I think it's a Bic ink. It has to be blue, and the ants will follow that because it's very similar. The chemical structure of that ink is very similar to the pheromone hormone that ants will follow. Anyhow, lock and key, you could have lots of chemicals that have pheromone-like activities for various different creatures, but only one will have the right signature that will fit the lock and bind into that site and trigger all sorts of happiness. So there you go. And so summary, unique combinations of 20 amino acids gives rise. Now the video said 21. I'm not trying to sell you or short you on something. There is a cysteine derivative, a slightly chemical alteration of one of the 20 amino acids. It's a unique case. We're not going to worry about it. So right now, 20 amino acids give rise to proteins, the chief building block of physical structures that make up everybody. So it's the keys on the keyboard. In other words, it comprises of all these different proteins based on these amino acids. Proteins perform a myriad of functions, from assisting chemical reactions to causing blood clots and bones and fighting disease and that sort of thing. So proteins are an essential dietary component. Now, a lot of you may be practicing vegans, or that you only eat plant material and that sort of thing, and then you're probably aware of the essential amino acids, because our bodies can't make certain ones of the amino acids, and we have to acquire it in our diet. And some plants, or eating plants, sometimes can't provide all of those amino acids. So another way of saying that is we look at complete, incomplete, and complementary proteins. Complete proteins have all the essential amino acids. Now you think about this. Let's say you're Henry Ford and you got an assembly line that you're building a Model T Ford, and we're happy. And all of a sudden we run out of tires. And once you get to the assembly line, all of a sudden there aren't any tires left to put on the car. So the whole operation, even though you got all the other inventory, because you don't have the tires to put on the vehicle, you have to stop the line, and or you just go without tires, and that means whatever you're going to have to live with it. So that's kind of what we're talking about here with incomplete. Um, so protein sources we have to worry about with plants. And so you can see various foods and things that we want to eat, like apples and almonds and rice and lentils. You can see which amino acids and things they may or may not have. So milk seems to be pretty complete and that sort of thing. So complementary proteins are eaten together to yield all. So in other words, there's only certain numbers of amino acids that we can actually produce through a biochemical pathway to build them, and we use some of the amino acids that we acquire in our foods as building blocks for other things. But anyhow, the idea is, as you go through and you're building and getting all the amino acids that you need, so you have to make sure that you're including those in your diet for the ones that we can't manufacture on our own. So 20 amino acids make up all the proteins necessary for growth and repair. All of these amino acids are about half the essential for humans. They cannot be synthesized by the body, and so they must be consumed in the diet. Now, I use a little acronym, which is a really good, powerful memory tool. I'm dyslexic, so I use these things. The first letter of the essential amino acids is private, PVT, TIM, TIM, Hall, HALL, and you go through the list of amino acids, and that will show or indicate all the essential amino acids. If you're interested, it's there. So protein's function is influenced by its three-dimensional shape. Oh, you're going to get tired of me saying that. But the amino acids are the primary structure, and they're held together by peptide bonds or amino acid bonds. And formally, when we have to talk with our suits and white ties, we have to have that sort of thing. But the idea is a peptide bond is the fancy chemistry name for linking the amino acids together. The secondary structure starts to form, like the video was saying, with the hydrogen bonds in a sort of a cyclic pattern. You can see that, or coiled, or in this stepwise type of formation, and we call that the secondary structure. The tertiary structure is the folding of a protein in liquid. Now remember, we have to have that liquid environment, and the subtle charge changes of all the R groups and things start to take an effect, and miraculously it starts to attract to those weak charges and then collectively builds into your tertiary structure by folding, and that's the mature structure. And if it's destined to interact with two or more polypeptide or these other bulk folded proteins, we call that the quaternary structure. So that has, this is showing three, and they're all bound together, and that's quaternary. Now we can denature when you cook an egg, for an example. You can denature it, and it turns from that gooey, gloppy, clear material to white. Or you can beat them, and they'll also turn white. And anyhow, what you're doing is you're denaturing it. Now we can do that with heating things, changing pH, and all sorts of other stuff. But the idea is you're disrupting that natural folding, and it becomes disastrous and breaks and that sort of thing. And sometimes, most of the time, when you denature, it can never go back. It's like trying to uncook an egg because of randomness, and it's broken and that sort of thing. But sometimes it only temporarily damages, and if you bring the temperature back, for an example, sometimes it can come back. So some people have curly hair. Well, some people have hair at all. I mean, I'm almost bald now. As it seems like every day I'm pulling my hair. I mean, I'm losing hair. And so some have curly and some have straight hair. And really, that boils down to the amino acid sequence. Now, I'm sure some of you have ventured into a mall or someplace. You know, a mall used to be a collection of stores. I've got socks older than most of my students, and I remember malls and things. But you go by a hairdresser that was doing a perm for somebody, and that god awful sulfur smell. It's a sulfur containing smell, and I don't like it. But anyhow, it's pretty pungent. But it's the sulfur that that causes that linkage between your hair, so you can have chemically added, or you could have natural higher instances of an amino acid with sulfur, and it causes the hair to curl a little bit. And it's just a simple little thing, but sulfur tends to bond a little bit stronger than others. And you can add chemicals to make it curl if you want with a perm, or keep it bent, or whatever you want to do. But naturally, if you produce it has a lot of that sulfur interaction, you get the curly hair, and that's genetic for the most part. So a protein's particular amino acid sequence determines how it folds. Remember, in a liquid environment, a particular three-dimensional shape, the magic. The charges are weak, but collectively, when it binds and folds, it's pretty strong. The shape determines many of the protein's properties and what it's going to interact with. And not only the shape, but the charge. And when a protein shape is deformed, a protein usually loses its function. In other words, you denature it, and it is no longer going to have that shape that it normally would, and it's not going to work for you. So, anyhow, so enzymes are proteins. Now, enzymes are amazing. Enzymes can be utilized in a reaction to catalyze a reaction, and then once it's done, it can be reused and reused until eventually the protein breaks down. But for the most part, I just want you to know enzymes can be reused. So so the substrate can bind like lactose, and lactase is the enzyme. Once it fits into that particular lock and key fitting, in other words, the shape and charge are allow it to bind. What happens when it binds, the protein changes its shape just a little bit and puts pressure on that bond. And when it puts that pressure on the bond, the amount of energy now it takes to break it is greatly reduced, and boom, you get the galactose and glucose molecules being released. Now, everyone, you can take a paper clip and bend it back and forth, and that heating process weakens that metal. It's the I forget the name of that. At some point, you break down the atoms between there. It's sort of what's doing it. You're weakening, you're putting pressure on it, and so you can bend back and forth that paper clip, and eventually it doesn't take hardly any energy to break it. And that's kind of the analogy I would use. It puts pressure on that bond once it binds into the active site, and puts a little bit of pressure on it. It doesn't take much now for the energy to break it, and that's greatly what we're doing. It's a catalyst. In other words, it's asking it to bind, and it helps facilitate. Now, the energy it takes for the enzyme to do this is built in. It's structural. So when it binds into the site, it starts to bend, like in this case, the lactose, and puts a little bit of a bind on that bond, and it's just easier to break it. And so that's reduces the energy it's requiring to do that. And I'll show you a plot that proves that. So chemical reactions occurring in the organisms can either release energy or consume energy. In either case, reactions need a little push. This is sort of the discount. It's like finding a coupon. I said it that way, coupon, but a coupon on the internet, and gives you a discount. Well, we like discounts, and the enzymes are really sort of like discounts, because it helps reduce the amount of energy or the cost when you utilize it, when you check out, or when the substrate binds the active site. And we call this the activation energy. The enzymes act as a catalyst by lowering the activation energy, and what that means is fancy gobbledygook. Here's the substrate. When it binds the receptor, normally, in order to break that, we have to have a huge amount of energy to break it. So we heat or do whatever it takes to create the energy. But if you add the enzyme and have the substrate bind into that enzyme active site, it puts enough of a strain on that bond. The amount of energy it takes now is greatly reduced. See this high? This is without the enzyme. This is with the enzyme. And the amount of energy it takes is greatly reduced. So it's like they applied the coupon here, so you got the discount. So you're not paying as much. And if you do this millions and millions of times, well, that's a lot of savings of energy. And that's how our bodies actually can sustain itself, by doing these sorts of things. And I hope you followed me on that, because that's really important. Don't tell anyone that I said coupons, okay? This was just a way to illustrate that without an enzyme, the cost is high to break it in terms of energy, but you reduce that amount of energy with the enzyme, because it puts a little bit of a bind on that bond between the two and whatever. And or it can save energy by building things too. It's not always just breaking, it could be building, and it puts them in such close approximation, close together, that it will be easier or less expensive to make it. So it works both ways, but I just wanted to illustrate that. I hope you followed me on that. Enzymes are proteins that help initiate and speed up a chemical reaction. They aren't permanent or altered in the process, and they can be used again and again and again. And so there you go. Well, this concludes. I know you're probably looking at your watch saying, "Oh my God, is he ever going to shut up?" So I'm going to stop here and pick up next time with the last part. It won't take very long. The last part is the finishing cherry on the top of all the wonderful things that you hopefully pulled out of this section. And so I'll see you next time. Please stay healthy so that I can torture you again with part 4.